All right, it's my pleasure to introduce our first speaker today. Uh, Dr. Arun Venugopalan is an assistant research professor at the Gulf Coast Aquatic Health Laboratory, which is part of the Global Center for Aquatic Health and Food Security at Mississippi State University. Arun holds a bachelor's degree in fishery science from Kerala University of Fisheries and Ocean Studies in India, as well as a master's degree in aquaculture from the Central Institute of Fisheries Education in Mumbai, India. He earned a PhD in infectious disease with a minor in computer science from Mississippi State University's College of Veterinary Medicine, specializing in aquatic virology. And he also completed a postdoc training at Woods Hole Oceanographic Institution in Massachusetts, focusing on the role of viruses in shellfish cancer development. So take it away, Aaron.
Thank you, Nick. And this is actually a really good moment for me. I actually would like to thank Nick, Matt, and all other Megan, of course, who kind of started this program because there was a time this started when the COVID hit, and I was a PhD student at that time. We did not have much opportunity to present or travel, and AFS FHS section really stepped up and created this program. And I was so I was with Dr. Matt Griffin and Dr. Larry Hansen doing my PhD, then did a postdoc in Woods Hole. Finally, I'm back on the Gulf port establishing a new aquatic animal health lab here. So I'm happy. So finally, I'm starting my own lab, and it's pretty new. It's just been 1 year. It's a new place, and we had a lot of construction and everything. So our equipment, so almost years we are a functional lab right now. But we do have more things coming in. I would like to introduce my research associate Adithya Bindhu Sreekumar, who will be joining us shortly after me. Then, of course, our team, Dr. Stephen Reichley, Mark Lawrence, and Beth Peterman, and myself. So we are the team trying to establish a new lab on the Gulf Port. So coming to the lab, especially to the coast, Mississippi coast. So we do have a fishery, especially with a large economic impact, about the 750 million US dollar. It has a nation's largest bay, which is marine mammal nursery, especially for sea turtles and dolphins. And we did had so I don't know if anyone know, the Biloxi here of the Gulf Port was known as seafood world capital. However, as you are aware, in 2005, Katrina happened. In 2010, deep water oil spill happened. Then we do have a freshwater incursion through the spillway. So once there was a really strong oyster industry or shellfish industry in general that has really declined and took a really hard hit with this all this natural as well as some man-made interventions. And currently, unfortunately, the entire oyster fishery is shut down, and we do not have a commercial landing of oysters anymore. Now, however, the state has really identified this as a priority area. In 2024, the state senate passed new regulation to allow new oyster leases across in the state, as well as they are making a large they already made around 55 million investment to revive the industry. So that is the time the Stephen and the team received a grant of 1.8 million from NOAA to establish a new aquatic animal health lab to support the coast and the coastal community, and especially shellfish industry and marine mammal program, and of course the shell finfish. And that's where I joined the team, and I'm currently located in the Gulf port. And when we ask what is our vision for our lab, as I already mentioned, this is Gulf Coast Aquatic Animal Health Lab under Global Center for Aquatic Health and Food Security. We do have, so I'm originally, I would like to call myself as a virologist. I don't know I'm really there, but of course, then we do. So ultimately, our focus would be to develop some One Health solution and how to support the blue economy on the coast, because coast, as I mentioned, has took a lot of hits, especially the fishermen community and the aquaculture farmers over the years due to the hurricanes and the oil spill and other things. So they do really need very strong support from the universities as well as the policymakers, which we are trying to develop that team. And as an aquatic animal health expert, this is what is my vision for our new lab. I'd like to focus on genomic epidemiology, especially with the long-read sequencing, nanopore sequencing. We already have a sequencer. Then One Health lab with the real-time sequencing of the pathogen, so that we do not have to wait a long time. Then. I'm planning, I'm in the process of establishing a high performance computing system where I would like to have our own bioinformatics servers, which is dedicated for the analysis or genome sequencing of novel pathogens or other new strains using the nanopore sequencing and other things. So of course, we do have a molecular diagnostic capability. One major focus research team would be microbiome, virome, interactome, and of course also the novel and emerging, how to characterize or identify novel and emerging pathogens in the aquatic ecosystem. So with an overarching goal of developing a sustainable blue economy over the course. With this, my research associate Adithya Bindhu Sreekumar would be starting with some of the work we currently doing.
A warm good afternoon, everyone. It's a great honor and pleasure for me to address all of you today. So, without any further ado, my presentation today centers on a persistent and an economically significant problem which the oyster farming or the commercial oyster farms had faced. JOD. So this JOD has had created a great impact, especially in the northeastern parts of the United States, which emerged during the late 1980s and created a severe mortality in juvenile Crassostria virginica. And first, I mean, we are very aware that oyster is one of the major aquaculture products in the United States. So, the disease has first emerged mysteriously with an unexplained mortality. I mean, it's a very severe mortality of more than 90% of more than 90% mortality in the juvenile oysters. And over the period of time, in the year 1999, it was described. I mean, the studies are conducted, and it was described as the Adenovirus crassostria was first described to be associated with the juvenile oyster disease. And later in the year 2005, it was described, or it was noted as the sole causative agent for the juvenile oyster disease. So, coming to the Allelosiovarius crassostriae. It is an aerobic gram-negative alpha proteobacterium, and which is a member of Rosiobacter clade. And the clinical signs which are associated with the juvenile oyster disease is the sudden cessation of the growth, which is followed by a flicking away of the periostrical growth on the upper wall, and the wall margins were noticed to be it could be observed as there could be an observation of uneven wall margins and the cup-like appearance on the lower wall, and the secretion of conchiolin in the on the surface, and lesions and retraction of the mantle. There were also the hemocytic infiltration, which is one of the primary indicators of the bacterial invasion. So, as a researcher, or as we are, when we were working with the when we were working with the traditional PCR methods, one of the major challenges which we face with the traditional PCR methods, one obviously it is the time-consuming one, and the other one is to differentiate between the biofilms, which may not be pathogenic always, and the other one is to differentiate it between the clinical isolates which are directly associated with the disease. So here comes the importance of our study. Another one is that I also want to give an idea about a study which conducted in the year 2021 in China, in which they could isolate a biofilm from the rock, which they could recognize as a reference strain of Allerosio varius grassosiae. So here comes the question that is that pathogenic to the oysters? So current PCR, as I have already mentioned, the current PCR methods cannot differentiate between the clinical isolates and the biofilm isolates, which may not be pathogenic to the oysters. So, and another question which may arise is that a new variant of the JOD bacteria? And since the US isolate does not have any complete genome, there requires a study or a deep understanding on it. So this present study mainly focuses on the qPCR assay, which can differentiate between the clinical isolates and the biofilm isolate. So the process or how the qPCR works will be described by Dr. Arun in the coming slides. And another challenge which normally researchers face when isolating or extracting the genomic DNA from the mollusks is the sticky DNA problem. So, like in plants, algae, or fungi, the mollusks, the mucopolysaccharides and polyphenolic proteins are the major components in the mollusk. These are the components which are mainly associated with the basal thread and the mucus. So during the extraction of the genomic DNA, they can even bind with the DNA, and it makes the extract more viscous, and thereby it may cause the difficulty to work with, especially during the pipetting. So another thing is that along with these two components, there are other pigments and contaminants which may also present in the mollusk, which may cause problems in the during the PCR or during the spectrophotometric reading. And there were various studies conducted for tackling this problem. And one of the most of the studies could substantiate that the DNA extraction using the CTAB protocol could tackle this problem. Mainly the strong detergent action of the CTAB can lyse the cell in a more efficient way and can remove or avoid the stickiness of the components like mucopolysaccharides and polyphenolic proteins. So as the time is a primary constraint, I can explain, or I can just give an idea about the CTAB protocol in a nutshell. So normally the tissue we will be taking for the DNA extraction first will be subjected for the CTAB extraction solution along with the proteinase K, and it is allowed for incubation overnight, and then it will be treated with the chloroform isoamyl alcohol, and later it will be subjected for the CTAB dilution solution, and then again we will be adding the NaCl in TE buffer, followed by the ethanol wash, and finally resuspension in the elution buffer. So here on next onwards, Dr. Arun will be explaining how PCR works and the other downstream applications. Thank you very much.
So that was the sticky DNA problem, and me and Matt always used to say that we should actually present all the problems we have so that others can listen to that so that they can avoid those problems. So this was a major challenge when we started working with the OSHA DNA, because getting the PCR working or genome sequencing is very difficult, because regular QIAGEN kits or other methods really doesn't work. Even the interesting thing is sometimes DNA quantity is good, but the inhibition is so can be very strong. We could see that. So that's why we got into the CTAB method. So that really works, and we did really optimize that CTAB and tested for PCR, and I was really successful for long read genome sequencing too. So that protocol is with us now. Uh, the next set would be, as you can see, as we earlier mentioned, there was a genome in 2016 they deposited for the ATCC type strain of for this bacteria, but they were incomplete. Of course, in order to fill the gaps, so what I did, I used the nanopore long read sequencing, and then I read a long read first assembly, then used the old short reads which they produced in 2016, and just to fix all those errors. And they actually missed two plasmids also, so I was able to get a complete genome without any gaps, as well as recover two plasmids. So now we got a complete genome of the type strain for this bacteria. Now, coming back to our question is, so the what happened was in 2021, researchers from China isolated some bacterial isolates. They grow and they did sequence it, and they were actually from the biofilm which grown from the rock. So our question was, okay, they are environmental isolates. Are they pathogenic to the oysters? Also, when they did the full genome sequencing and they uploaded to NCBI, and those genomes were completed, and NCBI now designate those isolates from the environmental isolates or biofilm isolates as the doesn't means they are pathogenic to oyster or they are really close to related. So I did a quick phylogenetic analysis. As you can see, this is our US isolates, which is originally, it's a clinical isolate originally from the oyster disease outbreak, juvenile oyster disease. And these are the isolates, biofilm isolates, and they are separate. So that means there is a taxonomic revision is required to really differentiate between this environmental isolates as well as the clinical isolates, and we are working more on this idea. And of course, the next step would be to develop a qPCR which can differentiate this environmental isolates and the clinical isolates from the oyster. So, as usual, this one was a bit tricky to find those regions which can really differentiate this environmental and biofilm, and it took a lot of time to really get into that. And of course, the inhibition with oyster DNA did not help either. So, but finally, I was able to put it together, and initially got 6 primers and prop set, then came down to 3, and even I had to adjust the primer concentrations almost. So if anyone really listening, and the Bustin, the who is originally part of the MIQE, he has a new publication and he has and a book chapter. He has really gave the step by step to optimize each component, and which really gives a really detailed steps to really tackle the difficult assays like this, and I was able to do it. And what you're seeing right now is the linear dynamics range of the assay determined by the tenfold serial dilutions. And as you can see, it has a 91.4% efficient. So this is without any inhibition. And we were able to get a sensitivity up to 10 copy numbers. So we were able to successfully validate this qPCR, which would differentiate the environmental isolates and the clinical isolate from oysters. So this is something which is ready for the to be implemented in the diagnostic labs. So with that, I would like to acknowledge my team. So myself and of course Dr. Rebecca Gas, Dr. Sharrett, then Abigail, Rachel, Roxanne, and Abigail Scrow from Woodsall and Massachusetts, where this work mainly happened, and I did some of the work here right now. Then. Of course, I would like to thank Dr. Stephen Reichley, Dr. Brett Peterman, and Dr. Mark Lawrence, who's currently I'm working with. And that's pretty much it. And I would like to take any questions.
Great. Thank you very much. Virtual round of applause for you and the team down there. Yeah, it's been fun watching you over the last 5 years or so present the work, and I'm so happy to hear that you're getting set up down there with your own lab and off to a great start. Folks, we have a little time for questions. If you have them, feel free to drop them into the chat, or you can unmute yourself and ask them directly. Either Aaron or Aditya would be great.
Looking at the chat.
Hey,
Okay.
hey, Arun, great job, man. And yeah, glad to have you back in Mississippi on the coast. Hey, out of curiosity, so my wife Shelly, she used to work down on the coast years ago, and there was a really heavy oyster like reef restoration program happening at that time. Is that still happening, or has that kind of like faded away? What is the status of that out near like Dauphin Island, that area down near Biloxi and Gulfport?
Yeah, it is still happening, and interestingly, it is really getting more active because, as I said, in 2024, Mississippi State Senate really passed a new law to give new leases for the farmers for the off-bottom culture as well as restoration. So they put 55 million already, and they're planning to put another probably another 50 million over the years. Also, their target for 25-26 is get a revenue for 25 million. So state is really, it's part of the governor plan also, so state is really putting a lot of investment to bring back the oyster industry. As I mentioned, currently it's closed. Harvest is not allowed because there isn't enough. But we are really going heavy on restoration as well as the farming also. So 2024-25, it's really getting into very active, and it has become a priority for the Marine Fisheries Department of Mississippi too.
Oh, that's great. I'll let Shelly know. She was there 2006 to 2008, so I'm sure we'll have to come down and see you, buddy, and see how things are going. But great job.
Yeah.
So we have a question in the chat here. Did you have any luck with the phenol chloroform extraction to resolve the sticky DNA?
So phenol chloroform, what we did was in the CTAB, we used the CTAB extraction solution, and yes, but phenol chloroform probably wouldn't work because there is I'm happy to share that paper because it has been tried, and I tried with the coagent. We basically tried multiple kits and multiple things. It looks like only the CTAB works, and again within the CTAB I had to really go ahead and do optimize certain things here and there to really get the quality. And especially for qPCR, we are still able to get away with some of the things, but when I needed the DNA for nanopore sequencing, especially 1 microgram DNA, that is a bit tricky. And if anyone knows, I even go for 4 times of ethanol wash, 70% ethanol wash, like really going crazy. Then ampere bead purification. So it really requires a little bit effort and time to get the clean DNA. PCR, I was, we with a decent effort, we can still get away with the PCR, especially with environmental master mix. words thanks to Matt Griffin. He found that magic. I still feel that is a magic master mix. So that would get away with a lot of PCR problems. But if you really want to get into the genome sequencing. So yeah, I'm happy to share that protocol if anyone really needs. So I'm not sure phenol chloroform really works. So CTAP protocol really works.
Sounds good. Thanks for the thoughts. And thanks again for the presentations.
Thank you.